everyone. Today we are going to talk about enzymes and we're also going to talk about metabolism. We are going to discuss cellular respiration. Enzymes are catalysts. What they do is they speed up reactions. They speed up reactions without getting altered and without getting used up. This is an enzyme right here. So let's say that you are speeding up a reaction, and this reaction started off with 10 enzymes. At the end of the reaction, you're still going to have those 10 enzymes. So they're not going to be used up, they're not going to be broken down. Another thing is that at the beginning of the reaction, if this is how the enzyme looks like, by the end of the reaction, it's still going to have the same shape. So the enzyme, structure-wise, is not going to be altered. And those enzymes at the end of the reaction can actually be reused one more time to start off the reaction again. So they can be reused. So the enzymes can be reused. Their shape is not going to be altered. They're not going to be broken down by the end of a reaction. Now, each enzyme has a site within it known as the active site. And each enzyme has a unique active site. So not all active sites look exactly like this. Now, what's going to bind to the active site is something known as the substrate. The substrate needs to fit within that active site. So not all substrates can fit within that active site. And once it binds, as you can see, it has to, one more time, it has to fit within that active site. Once it binds, the enzyme is going to go ahead and start speeding up the reaction. So if this substrate right here needs to be broken down, as you can see, once it binds to the enzyme is going to start breaking down that substrate, and you're going to get the enzyme unchanged, it's not going to be altered, and it can be reused if there are still more substrates that need to be broken down. So as long as there are still substrates, the enzymes are going to be reused until you are out of all of those substrates. Let's take an example. Let's say that this substrate right here that needs to be broken down is lactose. Lactose is the sugar found in milk. Now, to break down lactose, you need the enzyme that's going to help speed up that reaction of breaking down lactose, and that enzyme is known as lactase. You will notice that enzymes names end in ase. So lactase is the enzyme that's going to speed up the reaction of breaking down lactose. Now, lactose is only going to bind to the enzyme known as lactase because it has the proper structure to fit within the active site of lactase. As I said, other enzymes are going to have different looking active sites, and lactose is not going to be able to bind to those other active sites. It's not going to be able to fit. Once lactose binds to the proper enzyme, which is lactase, lactase is going to go ahead and it's going to start speeding up the reaction of breaking down lactose. Once it breaks down lactose, you get the enzyme, and if there's still more lactose, then the enzymes are going to be reused one more time until all the lactose within the body is broken down. Now, individuals that are lactose intolerant, what happens is those individuals do not produce enough lactase, or they don't produce lactase in the first place, and so what happens is the lactose is not going to be broken down because they're missing that enzyme, or they're not producing enough of it. So the process of breaking down lactose is nearly impossible because it would take forever without that enzyme. Remember, enzymes speed up reactions. And so if the reaction is extremely slow, it's not even going to take place. And so what happens is the lactose starts to accumulate because it's not being broken down, and it causes so many problems like bloatation, diarrhea, gas, etc. This is an image kind of showing you how the enzyme is going to fit within the active site of the enzyme, and it has to be a proper shaped substrate to be able to fit within that unique shaped active site of the enzyme. And once they bind, you can kind of see the enzyme kind of hugs the substrate, and the reaction can go ahead and get started. The way enzymes work is they lower what's known as the activation energy. What is activation energy in the first place? Activation energy is basically the minimum amount of energy that needs to be inputted for a reaction to go ahead and take place. Or you can think of it as the hurdle that needs to be bypassed for a reaction to take place. If you don't input enough energy and you don't bypass that hurdle or that activation energy, basically the reaction is not going to be able to take place. So let's say that you're starting off with some reactants right here, and you want to go through a reaction for you to get the final results, which are the products. If the reactants are here, those reactants, they will have to overcome this hurdle right here, which is known as the activation energy, for this reaction to go ahead and take place and for you to get the products that you need. And so this hurdle right here is known as the activation energy, or basically the minimum amount of energy that needs to be put in for the reaction to go ahead and take place. If the reactants don't make it up this hurdle, then the reaction is not going to be able to take place. So what enzymes do is they go ahead and they lower that activation energy, so it's going to take less time to reach the products. And that's how with enzymes reactions are sped up. And the reason why the reaction is proceeding pretty fast is because that activation energy is being lowered. So when the activation energy is lowered, then think about it this way: the reactants don't have to kind of go through or bypass a huge hurdle for them to reach the other side, the product side. So it's going to take less time. So if we take a look at this graph right here, so let's say here are the reactants, and a reaction needs to take place, for those reactants to start reacting together and for them to go ahead and give off the products. Now for this reaction to take place, as I said, those reactants, they need to overcome that activation energy right here. Now, this blue graph right here is showing you the activation energy that needs to be inputted for those reactants to get over to the other side and basically for you to get the products. Now, if this reaction is taking place without the enzyme, notice how big that activation energy is. Now, if we go ahead and add enzymes, what the enzyme is going to do is it's going to lower that activation energy. So see how huge that activation energy is? Once enzymes are added, the activation energy is going to be lowered. And so therefore, the reactants are actually going to be able to give off the products in less time because you've lowered that activation energy. It's basically a less of a hurdle now for those reactants to bypass, like to pass and get over to the other side. Enzyme names usually end in "-ase". Whatever comes before "-ase", which is the prefix part of the name, could give you hints about which substrates are going to bind to that enzyme, or it could give you hints about what this enzyme is going to do. So for example, if I say lactase, that name is giving me a hint that the substrate that's going to bind is lactose. If I say sucrase, then this enzyme is going to bind to the substrate known as sucrose. If I say oxidoreductase, that's an enzyme that's actually going to speed up oxidation and reduction reactions, which we are going to talk about in a few. If I say transferase, this enzyme is going to help transfer something, usually some functional groups like hydroxyl groups, like carboxyl groups, etc. If I say hydrolase, hydro meaning water, so that's an enzyme that's going to help break down things by adding water. So that process is known as hydrolysis. Ligase, it's an enzyme that's actually going to help stick things together. So stick things together, ligate them together. And usually that process needs energy, so ATP. There are many factors that can actually affect the activity of the enzymes, such as the temperature, pH, substrate concentration, and inhibitors. If any of these factors change, then the enzyme activity could either slow down or it could even completely stop. Enzymes work their best when they are placed in an optimal environment. Each enzyme has specific requirements. So if you take a look at the enzymes within your stomach, they have different requirements compared to enzymes within your blood. Why is that the case? Well, because the environment within your stomach, it's highly acidic, for example, compared to the pH of your blood. So the enzymes that are going to be within your stomach work their best when the pH is around 2, because that's the pH of your stomach. The enzymes within your blood are going to work best when the pH is set to around 7, because that's like the condition of your blood. The pH there is around 7. Now, if you go ahead and switch their places, so take the enzymes of your stomach and put them in your blood and vice versa, those enzymes are not going to function properly because you've changed the pH and therefore you've changed their optimal environment. Now, what happens when that takes place and enzymes are not functioning properly? The thing is, basically, the enzyme is actually going to denature. It's actually going to start breaking down. And when the enzyme starts to break down, of course, the reaction is going to be affected. And the reaction is going to slow down to the point sometimes the reaction could completely stop. And so if this is an active functional protein looking like this, and let's say you change one of those factors like the temperature, the pH, etc., the protein is actually going to break down. Let's go ahead and take a look at the factors that can affect our enzyme activity. So if we take a look right here at this graph, let's say over here we are inputting our temperature data, and then over here we are looking at the enzyme activity. Notice how the enzyme activity is the highest, meaning that this is where the enzyme is functioning the best. So this is like the optimal temperature up here for the enzyme. And so the optimal temperature, let's say it's around 37 degrees Celsius. So this enzyme works best when it's at 37 degrees Celsius. If you start lowering that temperature, notice how if the temperature is set at 20, the enzyme is still functioning, but notice how its activity is pretty low. Same thing, if you increase the temperature, so once, like as we said, the optimal is around 37, so if you increase the temperature to around 45, one more time, you notice that the enzyme activity is pretty low, to the point that if you increase the temperature way higher, or if you drop it, the enzyme activity could actually become zero. Like the enzyme is not going to function whatsoever. And so, as you can see, temperature is definitely a factor that can affect the enzyme activity. And what happens is when you, let's say, increase the temperature or you decrease it, why is the enzyme activity dropping? Because the enzyme, and enzymes are proteins, they are going to be denatured. They're going to denature, and therefore they're going to start inactivating. What's an example of temperature deactivating a protein? When you cook an egg, the white part of the egg, or when the egg is raw, it's the viscous part. That viscousy part of the egg is actually made out of albumin protein. When that albumin protein is heated, it's going to change from being clear viscous to becoming the white part of the egg. Now, why did it change from from clear viscous to the white part of the egg? Because you've heated it up, so you've increased the temperature, and the protein within within the viscous part of the egg could not handle such high temperature, so it actually started to denature. And when it denatured, it turns into that white part of the egg. Right here, another thing that affects enzyme activity is pH. You can see right here, the pH is graphed on the x-axis, and one more time over here, we're looking at the enzyme activity. Notice how this certain enzyme works best around pH of 5, which is pretty acidic. Now, if you go ahead and change the pH of, let's say, a solution, and you go ahead and make it more acidic, or you turn it into a basic solution, one more time, the enzyme is going to start deactivating, and you can see that the reaction is going to start slowing down to the point, again, the reaction could completely stop. If a reaction needs to be sped up, we said that we add enzymes. Now, if you want to speed up a certain reaction even more, what could be done is add more substrates, because the more substrates you add, the more those substrates are going to bind to the active sites of the enzymes, and the enzymes are going to work pretty fast to go ahead and speed up that reaction. So the more substrates you add, the faster the reaction is going to go. But there will come a time where the reaction is going to plateau. So the rate of the reaction is going to plateau out. And the reason why you see that plateau right here, because even though you're adding more and more substrates, if all you have are, let's say, 200 enzymes, and currently all the 200 enzymes are occupied by the substrates, and you go ahead and you add some more substrates, Well, the reaction cannot go any faster because all the enzymes are currently occupied by all of these substrates. So we say the enzymes are basically saturated. They're saturated with substrates, and so therefore you will notice at the beginning, the more substrates you're adding, the faster and faster the reaction is going, but there will come a time where now all your enzymes are currently occupied by the substrates. So even though you keep adding more and more substrates, that's it. The reaction cannot go any faster. It will plateau. Now, another thing that can affect the rate of enzyme activity is adding inhibitors. We've got two types of inhibitors. One type is known as the competitive inhibitor. They're called inhibitors because they're going to slow down or even stop the reaction completely. And the reason why they're called competitive, because they're actually going to compete with the substrate to bind to the enzyme. Remember how we said each enzyme has a specific site within it called the active site, and that active site has a unique shape, and only substrates that have the proper shape, they are the ones that are able to bind to the active sites. Well, what happens is there are some inhibitors that have similar shapes and they can bind to those active sites because they have, again, similar shapes to those substrates. So you can see right here, this competitive inhibitor competed against the substrate, and notice how it kind of got to the enzyme before the substrate did. So if this inhibitor got to the enzyme before the substrate, and basically it's now bound to the enzyme, notice how when the substrate comes to go ahead and bind to the enzyme, it's not going to be able to, because it's not going to fit anymore, because the active site is currently occupied by that inhibitor. So when those competitive inhibitors, they bind, what happens is the reaction is not going to proceed. Why is the reaction not going to proceed? Because the actual substrate never got to the enzyme. Actually, some of the painkillers that are taken to basically inhibit pain, that's how they work. Those medications could actually act as inhibitors, and they bind to the enzymes that will basically, those enzymes' job is to speed up the reaction of sending the pain signals to your brain. And so when those inhibitors bind to those enzymes, basically the pain signal is not going to be able to get to your brain. So they're basically going to inhibit the signal from getting to your brain. Another type of inhibitors is known as non-competitive inhibitors. Again, inhibitors because they're going to slow down and stop the reaction, but they're called non-competitive because they're not going to compete against the substrate. They're not basically trying to get to the active site. Those non-competitive inhibitors, they're actually going to bind to the enzyme, but at a different location. So if this is the enzyme right here, and here's the active site, notice how there's another site right here on the site of the enzyme. We call this site right here allosteric site, and the term allosteric just means different. So those non-competitive inhibitors, they bind to those allosteric sites. Okay, so what's the big deal if they go ahead and bind to those allosteric sites? Now, once they bind to those allosteric sites, what's going to happen is the active site is going to change shape. So it's going to be altered. And once it's altered, guess what? That substrate is now not going to fit. And so therefore, the substrate is never going to bind, and one more time, the reaction is going to be inhibited. So those are the two types of inhibitors. competitive and non-competitive inhibitors that can definitely affect the enzyme activity. They can slow down the reaction. They can even completely inhibit and completely stop the reaction from taking place. Now, to stop a reaction, we said that inhibitors can do that job, right? Whether they're competitive or non-competitive. Now, there is another way to stop a reaction, and that is known as the feedback inhibition. Now, what is the feedback inhibition? Let's say that you have a substrate, and that substrate binds to its enzyme, and the reaction does take place. And let's say that first reaction resulted in a byproduct. That byproduct binds to another enzyme, and you get a second byproduct, which binds to a third enzyme until you get to the end product that you are after. So sometimes actually to get to that end product, it takes several substrates and it takes several enzymes before we reach that end product. Let's say that this is a reaction happening within your body, and your body receives that end product, and the reaction keeps going, and so the end product keeps increasing, let's say, within your body. Now, we have receptors within our body that can sense how much of that end product do we have. And so once your body senses, because we have those chemoreceptors, for example, that can sense how much of a certain chemical we have, once those, let's say, chemoreceptors sense that we have a good amount of that end product, your body is actually going to want to stop the production of that product. So the product itself can actually bind to the enzyme, to one of the enzymes, and kind of act as a non-competitive inhibitor and bind to the enzyme and stop the reaction from taking place. So they can actually act as those non-competitive inhibitors and stop the reaction. So a non-competitive inhibitor could actually be the product itself and not a different inhibitor. So it could be the product itself, and the reason again why that feedback inhibition is going to take place, because your body has enough of that product and wants to stop the whole entire reaction, so it will bind to the enzyme and therefore stop the reaction from taking place, and that is known as the feedback inhibition. Now let's talk about two main reactions that take place within our body, and that is catabolism and anabolism, or you can call it catabolic and anabolic reactions. What are catabolic reactions? Catabolic reactions break down molecules. So if you have a macromolecule and you break it down into smaller, simpler molecules, that's basically a catabolic reaction. And usually catabolic reactions, when they take place, they release energy. We refer to that as exergonic, coming from the word exit. They're going to release energy. Anabolic reactions, they do the opposite. So anabolic reactions, you have simpler molecules and you basically combine them together to get a macromolecule, macro meaning large. And usually energy is inputted. How do we say energy is being inputted? We use the term endergonic, ender meaning inside. So the energy is being absorbed for you to get to that macromolecule. Anabolic and catabolic and anabolic reactions, they're basically reversible. They are reversible reactions. Now, what's an example? Let's say that you eat a piece of bread. That piece of bread has a macromolecule known as starch. So when you eat that piece of bread, you need to break down the starch within the bread. And so when you break down the starch, which is a macromolecule, into basically simpler sugars, that is going to be known as a catabolic reaction. and energy is going to be released when you are breaking down that piece of bread. Now, same thing. Let's say that you eat meat, and meat has protein. So you take your body is going to take the protein out of that piece of meat that you've just eaten, and your body is going to break down the protein into its simpler molecules, which are basically amino acids. So you break down the protein into amino acids. Again, that's a catabolic reaction, and energy is going to be released. Now, let's say that you have amino acids now in your body, and your body wants to build a protein. Your body's going to take those amino acids, input some energy, and it's going to build a protein. And so, as you can see, catabolic and anabolic reactions, they are basically reversible. When we talked about our catabolic reactions, we said that catabolic reactions are exergonic, meaning that they release energy. And when we talked about anabolic reactions, we said anabolic reactions are endergonic, meaning the reactants absorb energy for you to get the products. Now, what does it mean, exergonic? One more time. What does it mean when I say energy is being released? It means that your reactants are actually losing, okay? They are releasing, or you can think of it as they are losing energy for the reaction to take place. So if we go ahead and take a look at this graph right here, which is graphing time and free energy of molecules, and let's say here are your reactants. As we said, for a reaction to take place, those reactants need to overcome that activation energy, for the reaction to go ahead and take place and for you to get the products. Now, notice how those reactants, as they were bypassing that activation energy, and let's say the reaction did take place, and here are the products down here. Notice how the products have way less energy compared to the energy that the reactants started off with. So one more time, here is the reactants, and this is how much energy they started off with. Notice how high it is. The products, notice how the products have way less energy. Why do the products have way less energy? That tells you that this reaction is exergonic. This tells you that energy was actually lost or released by the reactants when they were reacting together to give the products. And so because energy was lost and released, those products have less energy compared to the reactants. Now, let's take a look right here at this reaction. So let's say that here are, one more time, my reactants, and they are starting off with some energy, okay, and here's the energy level right here. And again, for this reaction to take place, activation energy needs to be overcome for us to get to the products. Now, here are the products. Notice how the products, their energy level is higher compared to the reactants. What does that tell you? It tells you that those reactants actually had to absorb or trap some of the energy, some of that activation energy that they have to overcome for the products to basically for the products to be achieved. And so that's why the products actually have more, we call it free energy. Okay, so that energy that we've been talking about, we refer to it as free energy. So the products have actually more free energy compared to the reactants. One more time, because the reactants had to absorb energy. So when the reactants absorb energy, the products are going to have more energy, and therefore it's known as endergonic reactions. When the reactants had to lose energy, That's known as exergonic. And when the reactants lost energy, basically the products are going to have less energy. We've already discussed how catabolic reactions release energy and anabolic reactions absorb the energy. So when catabolic reactions release the energy, energy is released in many forms. It could be released in the form of heat. So heat could be one of it, and ATP. So mostly ATP is the energy that's going to be released. ATP stands for adenosine triphosphate. Tri meaning it has three phosphates bound to each other. Now, when ATP breaks down, it breaks down into adenosine diphosphate and an inorganic phosphate. And when ATP breaks down, it actually releases energy. Di meaning two phosphates, and here is this inorganic phosphate that was broken off. And actually it could, that inorganic phosphate could actually bind one more time to the adenosine diphosphate and basically turn back into ATP. So the production of ATP and the breaking down of ATP is reversible, and the cycle could keep going. So right here, ATP, which stands for adenosine tri, which means three phosphates. So you have three phosphate molecules bound to each other. That's ATP. That's the energy that we release. Now, ATP is usually broken down, and when it breaks down, basically one of these phosphates, if you see right here the bond, that bond is going to break down. And so when that bond breaks down, you get an inorganic phosphate. You actually get energy from breaking down that bond, and you get adenosine diphosphates, which means two phosphates are bound to each other. Now, once you get ADP and that inorganic phosphate can actually bind one more time to that ADP right here, and that process is known phosphorylation. So phosphorylation is whenever you're adding a phosphate. So right here, ADP is about to become phosphorylated because that P is going to bind one more time to it, and we can get our ATP back, our adenosine triphosphate. And as we saw in the previous image, that reaction is basically going to cycle, and we can get ATP, break it down, and then basically rebuild it by phosphorylating the ADP. Now, the reaction oxidation and reduction, those are actually two reactions that take place a whole lot within our body. Oxidation is basically the loss of electrons. Reduction is the gain of electrons. We usually use the mnemonic oil rig. So oil rig, if we look at this mnemonic, oxidation is loss, reduction is gain. So oil rig. Loss of what? One more time. Loss of electrons. Reduction is the gain of electrons. So a lot of the reactions that take place within our body, for this reaction to actually go ahead and take place, you usually have a reactant, and this reactant loses some of its electrons, and another reactant gains those electrons. So the reactant that lost its electron, we say that it's been oxidized. So you can see right here, reactant A lost some electrons, so it got oxidized, and reactant B accepted those electrons and gained those electrons. So reactant B got reduced, meaning it gained those electrons. And so this reaction right here is known as an oxidation reduction reaction, or for short, we could just say a redox reaction. So you can see right here one more time. Let's say reactant A, which is that yellow one right here, has some hydrogens, and hydrogen element contains both protons and electrons. So let's say this reactant lost some of its hydrogens. What does that mean? It means that it lost some of its protons and some of its electrons. Now, those electrons could actually be accepted by what we call a coenzyme. This coenzyme, its main job is to basically accept those electrons that are being lost. One coenzyme is NAD positive, and again, it's going to accept those electrons that are being lost. So reactant A, what happened to it? It got oxidized because it lost its electrons, and that coenzyme basically gained those electrons, so it got reduced. So you can see right here, it gained those basically protons. We call it protons because it's carrying a charge. So since it gained it, we say that it's been reduced. If it lost those electrons, we say it's been oxidized. And again, that's the redox reaction. So a lot of the reactions happening within our body actually are redox reactions. Now, as we said, ATP, it's adenosine triphosphates, and when we break it down, we get lots of energy, especially, as we said, like when you break the phosphate bonds and you take off one of those phosphates and you turn it into ADP, that actually releases a whole lot of energy. And so ATP is actually super important for us to produce and to use. Your cells need ATP for them to function properly and for the reactions to take place. Lots of reactions also need ATP. So for us to produce a good amount of ATP, we actually go through what's known as the aerobic metabolism, or you can refer to it as aerobic cellular respiration. That's when we break down food that we consume or macromolecules that you already have in your body, and you break it down and you get 30 to 32 ATP. Why am I saying food that you already have in your body or macromolecules that you have already in your body? Because let's say you haven't eaten in a while, but you have storage of food or storage of fat, you can actually break down your storage and still get a good amount of ATP. So we say that for every glucose molecule that we break down, we can get anywhere from 30 to 32 ATP molecules. And of course, in our body, we're not just going to have one glucose molecule. We have a lot, and it doesn't have to be glucose. You can actually break down other things than glucose. You can break complex carbohydrates. You can break down lipids. You can break down proteins and also get the 30 to 32 ATP molecules. Now, the reason why it's called aerobic, though, because we need oxygen to be able to go through that aerobic cellular respiration. But if your body doesn't have enough oxygen, we go through something known as anaerobic. Anaerobic meaning you don't have enough oxygen. So we call it anaerobic metabolism, or you can actually call it fermentation. So fermentation is basically anaerobic metabolism, and when we go through anaerobic metabolism, we only produce 2 ATP. So you can see the huge difference that between aerobic and anaerobic. Aerobic produces 32, anaerobic only produces 2. Now, when do we go through anaerobic metabolism? As I said, when your oxygen levels are low, for example, when you are working out. So when you start working out, even though when you're working out, your respiration rate is going to increase, and you're going to be intaking more oxygen, it's still not going to be enough, and your oxygen levels are going to start dropping, and it's not going to be enough for you to go through aerobic metabolism. So your body switches to fermentation, and that's when you start producing lactic acid. So part of why when we are working out, our muscles start to ache, well, of course, it has to do with the exercise, but another thing is it has to do with the lactic acid building up in your muscles because you are going through anaerobic metabolism. And anaerobic metabolism is going to produce 2 ATP, which is not a whole lot, but it is going to be enough just to kind of keep us going. But then, of course, your body is going to need to rest, and you're going to have to intake more oxygen, and your body is going to go back to the aerobic metabolism. Aerobic respiration needs lots of oxygen. Anaerobic respiration or fermentation doesn't need a lot of oxygen. So when we are working out and our oxygen production starts to drop, our body is going to go through fermentation to produce the ATP. The first step is glycolysis, which is the breakdown of sugar. But we are going to take a look later on and talk about how it's not just glucose that we break down. We can break down other things like lipids and proteins to produce ATP as well. It's just that the easiest thing to break down is actually glucose. So that's why we usually talk about glucose, but again, it's not the only source that we break down to make ATP. So glycolysis, once we break down glucose, we do produce energy, ATP. We produce about 2 ATP molecules, and we also produce something known as pyruvic acid. Now, during fermentation in humans, pyruvic acid is turned into lactic acid. And the lactic acid, as it starts to accumulate in your muscles when you're working out, it is going to cause pain. Now, other than humans going through fermentation, what other organisms also go through fermentation? Yeast. When yeast goes through fermentation, it does go through glycolysis and it produces ATP, and it converts glucose into pyruvic acid, but then it doesn't convert it into lactic acid. That's what happens in humans. Yeast turns the pyruvic acid into alcohol and carbon dioxide. And that's why we use yeast to produce beer, since it produces alcohol during its fermentation process. And that's why when we add yeast to bread, the bread comes out puffy, because when yeast ferments, it produces carbon dioxide, and so that gas is going to help rise your bread. That's why if you don't put any yeast, the bread comes out very, very flat. Now, aerobic respiration needs lots of oxygen. So when we are not working out and we're just gonna, resting, etc., and we have enough oxygen within our system, we still go through glycolysis. We break down glucose, we get 2 ATP, we break it down into pyruvic acid, but then we move on to a second cycle known as the Krebs cycle, or we can call it the citric acid cycle. During that process, carbon dioxide is released, and that's actually most of the carbon dioxide that we exhale comes out from the Krebs cycle. We produce another 2 ATP molecules, and during the process of glycolysis and the Krebs cycle, we go through the oxidation reaction. And that's when, as we said, oxidation is the loss of electrons. So during the process of glycolysis, electrons are going to be lost, and same thing with the Krebs cycle, electrons are going to be lost. Those lost electrons, they're going to be picked up by NAD and FAD, and when an NAD and FAD gains those electrons, they're basically going to become NADH and FADH2. So NADH and FADH2, you can think of them as electron collectors. They collect all of these electrons lost by glycolysis and the Krebs cycle. Now, what happens to all of these electrons that are now carried by the NADH and FADH2? These electrons, they're going to be used in the third cycle known as the electron transport chain. So during the electron transport chain, all of these electrons are going to be used to produce ATP. And I'll show you a picture explaining how this takes place. So during glycolysis, as we said, about 2 ATPs are produced, and then during the Krebs cycle, another 2 ATP molecules are produced. So far, that's a total of 4 ATP molecules produced. As we said, during the aerobic respiration, a total of 32 ATP molecules are produced. So that means to reach 32, we need to produce 28 more. And so 28 ATP molecules are going to be produced during the electron transport chain. So 2 from glycolysis, 2 from Krebs, and 28 ATP from the electron transport chain. So as you can see, the electron transport chain is extremely important in the production of ATP. What else is going to be produced and released from the electron transport chain? Water. So water is going to be produced. So when you think about it, aerobic respiration is really when we are inhaling and exhaling, and that's when you inhale the oxygen. So you intake the oxygen, you start breaking down the food in your body, and then when you exhale carbon dioxide that's coming out from the Krebs cycle, and you also exhale some water vapor that's coming out from the electron transport chain. This image right here kind of shows you that we don't just break down glucose, even though, again, glucose is usually what we talk about because it is the easiest to break down. But as I was saying, we can break down lipids, which are our fats. We can break down proteins. And as you can see, like when you break down lipids and you get some glycerol and some fatty acids, those byproducts that you get are either going to enter into the glycolysis cycle, or they're going to enter into like the step right before the Krebs cycle. But as you can see, you can break down other things other than sugars, which, like, glucose. You can break down things like proteins and lipids and still produce ATP and still go through basically the aerobic respiration process. Let's take a look at the glycolysis process together. Glycolysis takes place inside of the cell, specifically inside of the cell's cytoplasm. Glycolysis is anaerobic, meaning that it doesn't really need a lot of oxygen for glycolysis to take place. If we have a glucose molecule right here, and those are representing the carbons. So glucose has 6 carbons. Glycolysis, the very first thing that needs to happen is the investment of 2 ATP molecules. So 2 ATP molecules need to be used. The glucose is going to eventually split into 2 pyruvic acids right here. During that process, oxidation reactions are going to take place. So when glucose splits, electrons are going to be lost, which is oxidation. So those electrons that are going to be lost, who's going to pick them up? These molecules right here, known as NAD positive. and they're depicted by these shapes with empty spots to kind of tell you that those empty spots right here are going to accept those lost electrons. So right here, those negative circles are basically representing the electrons. So NAD positive, as you can see right here, it's going to accept those electrons that are lost. And when it accepts it, it's going to become NADH. So NADH are now carrying those electrons. So if you think about it, NAD positive is going to be like the oxidized state, and NADH is going to be the reduced state, because as we said, reduction is gain of electrons. Next, what's going to happen is 2 ATP for each pyruvic acid is going to be released. So therefore, 4 ATP molecules are produced. But what is the net outcome of ATP? The net outcome is only 2 ATP. How come? Even though you did produce 4 ATP molecules, you had to use 2 ATP at the beginning. And so therefore, the net profit, really, if you think about it, what is the net profit or the net outcome of ATP is just 2. Because I produced 4, but I invested 2 earlier. So technically, my net profit or my net outcome is 2 ATP molecules. So at the end of glycolysis, what are the main products that are going to be produced? The net outcome is 2 ATP molecules and 2 pyruvic acid molecules and NADHs. And those NADHs are basically your electron carriers. Then we move on to the Krebs cycle or the citric acid cycle if there is enough oxygen in our system. So if there is enough oxygen, we move on to the Krebs cycle. If there isn't enough oxygen after the glycolysis, we are going to start fermentation, where we convert our pyruvic acid into lactic acid. But if there is enough oxygen, as I said, we move on to the Krebs cycle. The Krebs cycle takes place inside of the cell, and it takes place within the mitochondria organelle. You can see that the Krebs cycle, you start off here with the pyruvic acid, and you can see that's a pretty complicated process as well, with so many different byproducts, many enzymes, many substrates, and the end of the day, the Krebs cycle, what is it going to produce? It is going to produce more ATP, and it's going to produce some carbon dioxide, and it's going to produce some electron carriers known as NADH and another electron carrier known as FADH2. So right here, here's the citric acid cycle or the Krebs cycle. These are the inputs. So you actually start off with some NAD positives and some FAD, and those, as you can see, they're kind of empty. Those are the ones that are actually going to, those are the coenzymes that are going to accept the electrons that are going to be lost. And then you get some acetic acid. That's a byproduct that you get from breaking down pyruvic acid. But the main outcome or the main outputs that you need to know is that once citric acid is done, once that cycle is done, what do you get? You get some carbon dioxide, and that's, again, most of the carbon dioxide that you exhale, or a lot of it, actually comes from the citric acid cycle. You get some ATP, around 2 ATP molecules. Those are just the steps. Those are not the numbers, so ignore these numbers right here. So you make around 2 ATP molecules, and then you're going to make or some of the electron carriers, the NAD positive and FAD, once they accept electrons, you are going to get the NADH and the FADH2, and you can see that they are carrying the electrons right here. So those are the main products that are going to be produced and released out of the citric acid cycle, or we can call it the Krebs cycle, which takes place inside of the cell's mitochondria. So you can see if someone has a problem with their mitochondria, or if their mitochondria organelles are degrading, and something is definitely wrong, you can see that the person is definitely going to have a problem with their aerobic respiration process, because the citric acid cycle takes place within the mitochondria. So as we said, from glycolysis, we produce 2 ATP molecules. Then from the Krebs cycle, we produce another 2, so that's a total of 4. And from aerobic respiration, as we said, we produce about 32 ADP molecules for 1 glucose. So that means that the electron transport chain is going to be the one responsible for producing the highest amount of ATP. If we want to reach 32, that means the electron transport chain is going to release about 28 ATP molecules. So you can see that the electron transport chain is really very, very important, as it's going to produce the highest amount of ATP. Now, what happens during the electron transport chain is that all of the electrons that have been carried by the NADH and the FADH2 from the glycolysis step and the Krebs cycle, all of these electron carriers, what they're going to do is they're going to go ahead and release those electrons that they are carrying. So they're going to lose those electrons. So as you can see right here, the NADH carrying those electrons are going to be released, and the NADH is going to return back to being NAD positive. So as you can see, like an oxidation reaction is taking place. As we said, oxidation is the loss of electrons. Those NAD positives, by the way, and FAD, those are going to be recycled back. So those NAD positives and FAD, they're going to be recycled back into the Krebs cycle and the glycolysis. And one more time, they are going to accept more electrons. And then eventually, those electrons, they are going to be released during the electron transport chain. And that's why we call it electron, because all of the electrons eventually are going to be released here. And it's called transport because you can see that those electrons are actually going to be transported from one complex to another. We're not going to go into details about these complexes right here, these complexes that you see right there. But basically, those electrons are going to be released. At the same time, the inside of the mitochondria, which is right here, the matrix, there are some hydrogen ions. Those hydrogen ions, they are going to be released from the inside of the matrix of the mitochondria to that intermembrane space, which is that space right here between the inner membrane of the mitochondria and the outer membrane of the mitochondria. When this happens, and the hydrogen ions, or we call them protons, are basically released from the inside of the mitochondria to that space right here. So basically the hydrogens are moving from here, the matrix, to that space right here, which is the intermembrane space. That's going to cause a lot of hydrogens, or a lot of hydrogen ions or protons to actually get concentrated in this region right here. Once you have a high concentration amounts of hydrogen ions right here in that inner membrane space, the hydrogen ions are going to start coming down this enzyme right here, known as the ATP synthase. As they pass through that enzyme, and the enzyme name kind of tells you what it's going to do, ATP synthase, meaning that this enzyme is going to help speed up the reaction of producing basically energy, ATP, as more and more hydrogen ions come down that ATP synthase, the more and more ATP gets produced. And that's how your electron transport chain is going to make the highest amount of ATP. So just to kind of recap what happens, or the most important steps that takes place during the electron transport chain is, one more time, those electron carriers, the NADH and the FADH2, they are going to release the electrons that they are carrying. Once those electrons are released, a bunch of protons, hydrogen protons, are going to move from the inside of the mitochondria matrix, and they're going to move to that inner membrane space. Then those protons, they are going to come down this ATP synthase right here, this enzyme. As they come down that ATP synthase, lots of ATP molecules are going to be released. In the meantime, there is some oxygen inside of the mitochondria matrix. That oxygen is going to accept some electrons and some hydrogens, and that's how we make water. So the main products that will come out from the electron transport chain is water, and that's the water vapor that we are going to exhale. Lots of ATP molecules, about 28 of them for each glucose that we've broken down. About 28 is going to be produced, and the main enzyme that's really going to help with that ATP production is the enzyme known as ATP synthase. So as you can see, the electron transport chain is extremely important in producing the ATP that our cells need, and basically our body needs. If anything goes wrong with the electron transport chain, let's say the ATP synthase is not functioning properly, let's say the electrons are not being released, or let's say the protons are also not kind of coming down that ATP synthase. If any of these steps basically gets stopped for whatever reason, then the whole electron transport chain could basically come to a halt, and your ATP production will stop. And if the ATP production stops, then we are not going to be producing enough ATP. Because if you think about it, glycolysis only makes 2, and Krebs cycle only makes 2. And we cannot survive on 4 ATP molecules. And so therefore, actually death can occur in such a short period of time if our ETC is not functioning properly.